Hi, my name is Luke Thompson. I'm an associate research professor with the Northern Gulf Institute at Mississippi State University and the Atlantic Oceanographic and Meteorological Laboratory in Miami, Florida. I'm going to give a final presentation about my project assessing ecosystem responses of Gulf of Mexico communities to OA using environmental DNA. First, I want to acknowledge my postdoc, Sean Anderson, and my co-PIs, Chris Kelbel and Leticia Barbaro, who contributed to this work. Marine plankton perform critical biogeochemical roles in the ocean, including primary productivity, nutrient cycling, and carbon export to the deep ocean. They can also be used, because of their important role, as indicators of environmental status. As we all know, ocean acidification is a major threat to the world's oceans in various ways, including to calcifying organisms. And how plankton respond to OA is really critical. We can also use the organisms in the ocean as indicators of ocean acidification. The Gulf of Mexico is critical to the American blue economy, but despite this, it has not been studied extensively to the extent of other major ocean bodies. Most of the Gulf of Mexico and the deeper regions are unsampled. And a spatial survey that included both biological and physical and chemical measurements, including ocean acidification, would provide a much richer picture of the ecosystem and how this ecosystem will respond to ocean acidification and other stressors. The picture on the right is from a paper by Emily Osborne and shows the various drivers of ocean acidification in the Gulf of Mexico. On the right is a simple phylogenetic tree of different eukaryotic phytoplankton species, including some of the major types and some of the types that may be influenced by ocean acidification, such as coccolithophores, foraminifera, pteropods, and other mollusks. So in this work, we set out to address a few research questions and also provide resources to various research needs. So one of the main questions is, how do different groups of plankton respond to ocean acidification as revealed through basin scale patterns in plankton DNA diversity paired with physical and chemical oceanographic measurements? And as plankton adapt to ocean acidification, either through selection or among standing genetic variation, how will this adaptation feed back on the biology and biogeochemistry of the marine ecosystem? And some of the needs that we are addressing is providing measurement of bacteria, phytoplankton, and zooplankton community structure, which are level 2 activities that link the core OAP measurements to these biological ecosystem components. We're also developing biological indicators of ocean acidification, which will have various uses. So on the GOMECC-4 cruise, which took place in 2021, we collected eDNA at 54 out of 104 sites during this cruise. We sampled at the surface, the deep chlorophyll maximum, and near the bottom at each site. And we did triplicate filters or samples at each depth. We then performed metabarcoding of the 18SV9 region to identify protists and also metazoa. I will not talk about the metazoa in this talk. And the 16SV4V5 region to capture bacteria and archaea along the transects, which span latitudinal and longitudinal axes, as well as depth from the surface down to the bottom. A quick overview of the eDNA analysis process. We collect a sample, we filter it onto a filter. In our case, we used Stereovex filters. We then perform DNA extraction back at AOML, prepare for sequencing. That's a 2-step PCR process that we use. We do the first at AOML and then the second one at Michigan State University sequencing lab. And then we get the data back and we perform analysis at AOML using our pipeline called Tourmaline, which uses QIIME2 and some other things. And I'll talk about that in a minute. So Sean Anderson was the main person on this cruise on the Ron Brown. And here he is sampling water from the CTD, collecting it in Whirlpack bags and bringing it back to the lab to be filtered using a peristaltic pump through Stereovex filters. The Stereovex filters are shown on the left. You can see some biomass that's accumulated on the filter during pumping. You can also see the QR-coded labels that we designed for this study that allow us to easily track samples when we extract them. On the upper right is shown the Kingfisher magnetic robot that we use to do a magnetic bead extraction method. And on the lower right is Sean working in the lab to run some gels to verify that we got amplification of the DNA. So when there's eDNA in water, it can come from a variety of sources as shown on the left. But ultimately, we extract that DNA into a tube, and then we can perform different analyses on it, whether quantitative or total shotgun metagenomics, or in this case, metabarcoding. So today I'm just going to talk about metabarcoding data that we did, and in the future we would like to do metagenomic analyses and maybe even some quantitative PCR on these DNA, which we can. We have leftover DNA. So in the metabarcoding, we targeted two different groups, or two different major groups, bacteria and archaea with the 16S gene, and protists and metazoa with the 18S gene. And then we, when we got the sequence data back, as I mentioned, we used tourmaline, which is a workflow that uses QIIME2. We assigned to these sequences that are called ASVs, or amplicon sequence variants, to public databases, PR2 and Silva. We then assigned those to functional groups using an existing schema that was developed for the Tar Oceans expedition, and then performed additional analysis in R using a variety of statistical packages that I'll talk about. In total, we have 472 seawater eDNA samples that were sequenced, and we got back over 9 million 18S DNA sequences and over 13 million 16S sequences. And again, 18S and 16S are shorthand for the 18S and 16S ribosomal RNA gene. We get DNA sequences from those. So what did we find? First thing that we did was we realized that we had a lot of samples from different depths, and in some cases what we were calling the deep sample was still quite shallow because of the shallow nature of the Gulf of Mexico in certain parts. And so what we did was we clustered all of the data, both 16S and 18S data, here I'm showing 18S, into clusters. And we identified that 3 was the optimal number of clusters using a gap statistic that I'm not showing here, but it will be included in the supplemental. And so we have 3 clusters. The cluster 1 is in red. I'm looking at the upper left here. Cluster 2 is in tan, and cluster 3 is in turquoise. And so cluster 1 and 2 are very close in depth, but they are very distinct still biologically. And then cluster 3 is more diffuse, but represents a deeper profile. And so we can then use these clusters to perform a variety of analyses. They provide a way to sort of separate out the samples. So looking at what's called alpha diversity or richness, or Shannon diversity, are different measures of alpha diversity. It's basically how much diversity is in a single sample. We see that there is an increase in relatively level protist diversity at the both richness and Shannon diversity level. And then when we look at beta diversity, which is shown on the right, we see a nice separation using a distance-based redundancy analysis. And this also allows us to see which physical variables are driving the separation of these different groups. So we have temperature at the surface, and we have DIC at depth, and various other variables that are shown on the right. With 16S, we also had three clusters, and they were essentially the same clusters for each sample. And we see, as opposed to the 18S, where the diversity is either more flat or actually decreases a little bit with depth, the 16S diversity seems to increase with depth. And we also see, separation based on this redundancy analysis, and it's a similar kind of pattern. But there's a lot more in the data that we'll see as we go forward. So one of the main things we wanted to do was look at what is the taxonomic composition of the data. And there's many ways to do this. There's many ways to slice the data. Obviously, you can look at each of those 472 samples individually, or as grouped by replicates, but here we're looking at 3 big bins. So we're looking at cluster 1, cluster 2, and cluster 3. So surface and coastal, near surface epipelagic, and deep mesopelagic. And so we see very clear patterns and shifts from the surface to the deep. In particular, a shift from lots of dinoflagellates to a little bit fewer dinoflagellates, and Rhizaria coming up, and Excavata coming up with the deepest samples like Diplonemia, and Polycystinia, the Rhizaria. Syndiniales are abundant in all 3 layers. Dinophyceae are most abundant in the surface and get less. And so this relative abundance data is very useful for understanding kind of the broad brush. What are the major groups out there? This is looking at protists. We can also look at bacteria and archaea. The alpha proteobacteria, such as SAR11, are abundant in all 3 layers, but more in the surface. Cyanobacteria also more abundant in the surface, in particular Prochlorococcus and Synechococcus. As we get deeper, we get Nitrosa pumilis and Marinin microbia, and some various other groups, in particular the archaea, that start to pop up in the deeper samples. So as I said, there's different ways to slice the data. We can also slice it by line. So this is going from Florida counterclockwise around through Mexico, from left to right. Here we're looking at richness and the relative abundance of different groups. And on the left is the 18S data, and on the right is the 16S data. And so there's some subtle patterns, nothing that jumps out dramatically, although there are a few exceptions like the Veracruz line, and a few others that we are going to dig into a little bit more in the manuscript. But there's a lot of resolution and different ways to slice this data. We also can identify different environmental drivers in addition to the sort of broad brush that I showed with the beta diversity redundancy analysis plots a few slides ago. You can also see the different variables that maybe are driving different groups. So we have these 5 groups shown in this slide, and how they correlate with different environmental variables using a partial least squares analysis. And this is looking just at the cluster 1, which is the surface samples. We also applied a general linear model, which can be used to predict the community and specific groups at different in different parts of the Gulf. And in particular, we can use this to predict what the community and what different groups their abundance will be where we weren't able to sample, right? So parts of the Gulf where we couldn't go, or future scenarios in particular. So what this is showing here is just how well, so what we did was we took 80% of the data and built the model, and then we took the other 20% and we tested how well those samples fit. So what we're seeing is about 20% of the data that we are plotting and fitting the predicted relative abundance and the actual relative abundance in the test set. And there's pretty good agreement, and so this gives us some confidence that the general linear model that we developed is going to be accurate. And so then we can apply that to the data set. And so there's a lot of data here, but what we're seeing is on the 18S on the left and the 16S on the right, how in this model, this general linear model, how the different groups, number one, how they correlated to the different environmental variables on the upper left of each plot. On the right, we have the relationship, the predicted relative abundance of the samples relative to DIC, temperature, and pH. And if there's a trend line, it shows whether the pattern was significant or not. And then on the bottom, we have a map where we have extrapolated, or rather interpolated, the abundance as a heat map of the different groups. So we have Dinophyceae, Syndiniales, Saginista, and Primnesiophyceae on the protist side, and on the bacteria archaea side, we have SAR11, Synechococcales, which is the Picocyanobacteria, SAR86, and Flavobacteriales. And so we see some really interesting trends with those different patterns, and we can then correlate that with things like DIC and pH, which I'll show in a minute. And then finally, we were able to develop indicator taxa of, or identify indicator taxa of, in particular, high DIC and low pH. So we chose these because they were the most informative for the models that we were developing. We also were able to identify around 10% or so of the samples that were outliers in terms of DIC and pH, so higher DIC. So DIC ended up being about greater than 2100 micromoles of carbon per meter cubed, and pH less than 8.0. And so on the protist side, the taxa, using an algorithm called index species, the taxa that were most indicative of protist, among protists, most indicative of this high DIC, low pH environment was Ostriococcus, a chlorophyte. And then among the bacteria and archaea was a number of different groups shown here that could have value in indicating this environment. And so this is the sort of all-in-one figure that we've highlighted. And so there's a legend on the right that you can read. And we're looking at these two really interesting groups of dinoflagellates, Syndyniales and Dinophyceae. And Syndyniales are these very abundant but poorly understood parasitic dinoflagellates. And so we can see how these two groups vary over DIC, temperature, and pH. In this particular model, the pH relationship for these two groups was not significant, but we do see both decreasing with increasing DIC, and then with temperature, the Dinophyceae decreasing, but the Syndyniales increasing. And so then one of the things we're doing, and this is the last piece that we're working on, is to incorporate climate models and emissions scenarios, so basically temperature and DIC, to predict the abundance of different groups sort of in 50 years, like say in 2050, so in 25 years, and in 100 years, or actually 75 years, so 2100. Basically, what is the current situation? What does our model say it is now? What does it predict for 2050? And what does it predict for 2100, given the data that we have from GOMECC 4 in 2021 using this general linear model? And so, for example, we predict that the Syndyniales, which is what's shown here, these parasites, will significantly increase in their abundance as temperature increases. And if we have a warming ocean, we expect to have more of these. And we can't totally predict, of course, what the implications of that will be, but that's a very useful piece of information to know that this group of parasitic dinoflagellates that's very abundant may increase in abundance in the future. And so to summarize the results, I'm just going to leave this here. I think I'm running out of time, so I'm going to leave this here and say that we have some really nice data on diversity of these different clusters, and we're able to take all that data and put it into a model of both unobserved regions of the Gulf as well as future scenarios. And so from this work, we have developed and generated the first map of planktonic diversity in the Gulf of Mexico that really spans the major regions plus depth profiles. Most of the work has been more on the northern Gulf of Mexico, and so we think this is the most comprehensive map of diversity in the Gulf of Mexico so far. We've identified major groups of protists and bacteria and archaea, and in these different depth layers and the environmental variables that drive them. And we've been able to highlight in factors influencing the distribution of Syndyniales, which are a highly abundant but poorly understood group. We've also identified indicator taxa of OA and built a model of Gulf plankton diversity and plankton communities in unsampled areas of the Gulf, and importantly under future climate scenarios. I also will mostly just leave this here. This is what we're proposing to do in the future, is to basically continue to do sampling on GOMECC 5 and with our partners at PMEL on the WACOA cruise, including, and we want to add whole genome shotgun sequencing that is the same kind of sequencing we're doing for BioGoShip, which is another project I'm heavily involved with, and to make that data interoperable across all of our projects, which we're well on our way, as I'll show in a minute, and then to model that additional data that we get from the metagenomic analyses. As well, we want to add POM, particulate organic matter sampling, and biooptical scattering, particle backscattering profiles, which will provide really useful data about other aspects of the plankton that we can't measure with omics methods, and also will enable us to calibrate BGC Argo float data. So there's a lot of synergy happening with those different measurements. Finally, I want to talk about our data management work. So we have developed templates for importing and inputting all of the GOMECC data and other cruise data into a single unified Google Sheet that has tabs for each of the different kinds of metadata and data, from studies to samples to prep to analysis, and then to submission to the SRA, or Sequence Read Archive of NCBI, as well as other repositories. So the link there, you can download the template there and learn more about it at our data management guide on GitHub. You can also read about or learn about our conversion software that was developed by Catherine Silliman, which allows uploading of data from this format to OBIS and publishing. So we have now published the GOMECC 4 data on GBIF and OBIS, so the Global Biodiversity Information Facility and the Ocean Biodiversity Information System. This is a major accomplishment. Took a lot of effort to get the data formatted in these formats, and it's now been accessed hundreds of times as part of this massive global data set. The links for those publications are at the bottom, and we're really excited about that. And finally, I'd just like to thank my co-authors, as shown on the left, and the OAP and NGI and OER for financial support. And thank you for listening, and look forward to continuing this work in the future.